Good morning everyone. So it is our first lecture, and today I will mainly focus on the basic introduction part of the microscope. I know that all of you are very familiar with microscope. This course is mainly for the biologists, so many of you are very much familiar with this microscope, how to use it. So what I want here, that first 1 or 2 classes we are going to discuss about the basic component about the microscope, and then we will discuss more about electron microscope. So whenever we are talking about a microscope, what it will come to our mind? So it will come something like that. So microscope is something which is enlarging, and it is helping us to visualize. But if I ask you definition, what is the definition of the microscope? So definition of the microscope is that is an instrument for observing the object which are very small to be visualized by the naked eye. As an example, bacteria, viruses, cell organelles. So microscope is an instrument which first discovered in 1592 and evolved over the time. We will discuss about this history of microscope in later slide. Now lot of time we mix up these two terms, that is called microscope and microscopy. So microscopy is a method where we are using this instrument called microscope to magnify extremely small molecules or samples and object that cannot observe with the normal eye. These objects are too small, which are not within the resolution range of normal eye. So microscopy is the technique or methods, and microscope is the instrument. Now here these two new terms are coming. One is magnify and another is that resolving power. So what is that magnification and what is called the resolving power? First we will discuss about the magnification. So magnification is the process of where we can enlarge the apparent size of the object, not the physical size of the object. This enlargement is quantified by the size ratio called optical magnification. So magnification is the ratio of the size of an image object seen under the microscope to the actual size of the object. So what does it mean? That we have an object and size of the object is 1 centimeter, and then we are using a lens and which is magnifying this small object, and we are getting a image of this object which is 10 centimeter. So how much we are magnifying that image? So if we just take the size of the image, that is 10 centimeter, and divided by our object size, that is 1 centimeter, we will get 10 times magnified image. That is called the magnification. We do not get any unit for this magnification because it will cancel by each other. And magnification we can tell that 10 times magnified means that 10x. So if it is 100 times magnified, we can say 100x, or 1000 times magnified, we can say 1000x. Now a compound light microscope is an optical instrument that uses visible light to produce a magnified image of an object or specimen that is projected onto the retina of the eye or onto the photosensitive surface of an imaging device. The word compound refers to the fact that two lenses, the objective and eyepiece or ocular lens, work together to produce the final magnification of that image. So when there is a compound microscope, in compound microscope we have two lens system, as we mentioned here, that we have one lens system, that is called the eyepieces, and the other one is the objective lens. So when we are calculating the magnification will be multiplication of objective lens magnification multiplied by ocular lens magnification. So how we are getting this formula that okay, final magnification should be multiplication of objective lens magnification multiplied by ocular lens magnification, that will come to the next or later slide. I also discuss about little bit about the resolving power. What is the resolving power? Resolving power is the power or ability of an optical instrument to separate the image of two objects which are close together. So resolving power of any lens or any microscope, it is like that if there is a two objects are closely packed, so it can magnify in such a way so we can see there is a two different object clearly. So it should give us a two clear object, it should not be merged like that. That is called the resolving power of a microscope. Now the optical microscope are often referred to the light microscope. Is a type of microscope which use visible light and a system of lens to magnify the images of small sample. Optical microscope are the oldest design of the microscope and are possibly invented in their present compound form in 17th century. Here there is a 2 point we should pay attention. One is the visible light and one is the lens system. In optical microscope, most of the time we are using two component. One is that radiation is basically depending on the visible light or the photon, and the lens we are using the glass lens because glass lens are good enough to converge the light and we can get an image. So in any kind of optical microscope we are using these two main things, that is the visible light and the glass lenses. That will be completely different when we will use electron microscope. That is the reason I told you to pay attention to these 2 points, that is the visible light and the glass lenses. So now it is always important to know little bit about the history of the microscope. So there are, even in 13th century, there are some images or some pictures by different painters is visible where monks are wearing the eyeglasses or they are using some kind of mercury compound and they covered the one side of the glass and they are using that as a mirror. But those informations are not documented that okay, in 13th century person X or Y is discovered this mirror or the microscope or the telescope. What we can find in the literature, that is Hans Leprasy, a Dutch spectacle maker who first patented a device that is called the telescope, that is called Hans telescope. So similar time frame in Galileo Galilei, who also built a instrument that is called the Galileo telescope, that is he is grinding that lens by hand. Here we can see the similar type. Johnson is another Dutchman. He also discovered the compound microscope. In similar time, Francisco Fontana, who removed the convex lens to concave lens in the eyepiece of the compound microscope. Now there is a huge discovery by Kepler in near around 1611, who discovered that law of refraction, and he also used the similar approaches where he moved the concave eyepiece and make it a concave lens and in his telescope, which is really helping to visualize in the better way. Okay, the next is Snell is the another professor from Leiden who discovered the law of refraction, and this is a very common and very famous discovery in the optics that we are using in the recent day optics also. So, here you can see there are couple of images are there. So, mostly those microscope or telescope we are talking about in general, but how we are implementing those things for biological study. So, here you can see that I already mentioned in 1590 there is a two spectacle maker, that Hans and Zacharias created the first compound microscope, and then Galileo Galilei discovered his telescope, and then the Robert Hooke is the first person whose information published in Micrographia journal, where he first described about the cell. He imaged those things using his own microscope. Now, another important person is Antony van Leeuwenhoek, who discovered the compound microscope, and this is his very simple microscope which he used to visualize the blood, yeast, insect, and documented those micrographs. Not only that, in the 18th century, 19th century, there are many famous scientists are involved with this optics, and they discovered many new things. And then there are two famous names, is that Thomas Young, and then Fresnel's law is very, very famous. So, we will discuss about all of this, how we implemented those things when we are getting a different types of diffraction pattern, then single slit experiment, double slit experiment. But this is a very brief history of the microscope, how the microscope is evolved gradually. Now, here there are couple of images are there which is showing you different types of microscope. Here you can see this is a compound microscope discovered by Hooke, and then this is the Leeuwenhoek, the single lens magnifying lens containing microscope, and this is a two eye pieces are there, which is a more sophisticated microscope than this compound microscope, and this is a modern days microscope where we can see there is a lot of complicated operation we can do with this modern days light microscope. So, as I mentioned previously that we should pay attention to two most important part. One is that in one particular case we have light source and we are using glass lenses, and other cases we are talking about that electron microscope. So, there are two major types of the microscope will be available. One is light microscope and another one is the electron microscope. So, light microscope is very simple, where we are using light microscope where we have the optical lens or glass lenses to converge the electron beam, and radiation will be either light or photon. Now, if you want to classify this light microscope, we can classify this light microscope in different classes. One is that polarized light microscope, then reflected light microscope, bright field light microscope, the next one, then phase contrast microscope, dark field microscope, and fluorescence microscope. These are very simple classification. We can get many more different types of microscope like that. If we just consider the confocal microscope, the confocal microscope will come under this section. So, there are many other light microscope will come, but all the cases common part is that the source or radiation source is light or photon, and we are constantly using glass lenses to focus the beam. In the other cases, we are using electron as a radiation source. How can we classify those systems? So, here you can see we have three major classification. One is TEM. TEM means transmission electron microscope. Transmission electron microscope, by name it is clear. So electron beam, if your sample is this one, electron beam will transmit it through your sample and then it will form an image at the image plane. So when electron beam is transmitted through your sample, that is the reason it is called the transmission electron microscope. In the other hand, if you look at the corner, the right hand side, it is written SEM. SEM means scanning electron microscope. What is that scanning electron microscope? Scanning electron microscope, it is, we have a sample here and then electron beam will focus in one particular region and then it will scan that sample, surface of that sample. So that is the reason it is called the scanning electron microscope. So it is something like that, you have a searchlight or torchlight and then you have a sample and you are focusing that torchlight in one particular area and then you are scanning that surface of that sample. So that is called the scanning electron microscope. Next it is coming that STEM. So it is scanning transmission electron microscope. So again, this is a very simple classification. There are many different types of microscopes, electron microscopes are there. If you just consider that, okay, analytical electron microscope, high resolution transmission electron microscope. So you can classify those things based on our use, what kind of use we have. But in some cases, some instrument we can use only for biological science research purposes, and other cases other electron microscopes are suitable for material science research purposes. So we have to know how we can use those electron microscope for biological science research purposes. Now here one common question can come, why suddenly we are talking about some microscope we can use it for material science purposes and some microscope we can use it for biological science research purposes. So here one thing we have to pay attention, that is called the electron microscope. Electron microscope is radiation is happening by electrons, and electron has a very strong energy, it can damage the biological science sample. So that is the reason for material science sample, most of the time it will be not a beam sensitive, so we can use any amount of electron beam. But here for biological science sample or any soft material, if we use a very strong electron beam, it will damage the sample. So those are beam sensitive. That is the reason there is a two different types of microscopes are coming into the picture. So here one very simple table is there, we do not have to pay attention to all of this, and again I am telling this is a very simple classification. We cannot classify all these electron microscopes here. Here the blue color, it is showing that transmission electron microscope, it is a transmitted electron beam, we can use it for biological science research purposes. And just below of that one, it is called the cryo-electron microscope. Cryo-electron microscope, it is again a same transmission electron microscope where electron beam is transmitted through the sample, but it is operating at cryogenic temperature. So that is the reason it is called the cryo-electron microscope. This cryo-electron microscope and transmission electron microscope, both we can use it for imaging of any biological samples. So now here, as I mentioned, that high resolution TEM or analytical electron microscope, most of the time we use it for material science research purposes or those kind of sample where beam damage is not a problem. Now scanning electron microscope can also classified into different classes. So here you can see this is a room. So there is a room temperature normal scanning microscope is there. We will discuss little bit about that room temperature scanning electron microscope also, but here you can see there are some classification is there, that is the field emission operated scanning electron microscope, environmental scanning electron microscope, low voltage scanning electron microscope, the cryo scanning electron microscope, all has different purpose and different usage. So here in this course we are mainly going to focus on transmission electron microscope, cryo-electron microscope and scanning electron microscope. Okay, now let us take a look what is the general design of simple light microscope. That will really help us to discuss about the overall architecture and anatomy of electron microscope. If we remember that light microscope, overall architecture of the light microscope, it will really help us to compare both the microscope side by side. Now any light microscope will get a base, and then there is a ocular lens or eyepiece, and then there will be a 3 objective lens. So this objective lens has different magnification. So here we are combining this eyepiece and the objective lens and we are giving the total magnification. So whenever it is required, we can change the objective lens and we can magnify something from 10x, 40x, 100x, 1000x or 200x. This is the arm which is holding the entire system, and sample will go somewhere here, and this is the light source. Light will come through this one and we can observe that sample here. Now there is a 2 knobs are there which is helping us to focus the sample. So coarse adjustment and the fine adjustment. So these are the 2 knob which is helping to focusing the sample. Now this is a little bit more complicated light microscope. What is that complicated light microscope? Where we have the lamp here and there is a intensity adjustment plane which is reflecting the light through the sample. Here we have the sample stage where our sample is here. This is the eyepiece. So light will come here, reflect and go through the eyepiece and we can observe the sample. Here the same way like this one, the objective lens, we have 3 different objective lens, and whatever magnification is required, based on that we can change this objective lens. Here we have the focusing knob which we can use the coarse and fine alignment focusing, and there is another 2 knobs are there which we are using to X and Y translator. So we can move the specimen from different position. Okay, so I discuss about the very simple light microscope, and this is a modern days optical microscope. So in this optical microscope you can see this is a very sophisticated microscope where this is the internal part of this optical microscope where you have that different types of objective lens. You can just use the combination of this eyepiece and different types of objective lens and get a different kind of magnification. This is the stage where you have the specimen, it's the condenser, and the light source is coming here and passing through that sample. So the transmitted lamp is located here. This is the focusing knob which is helping us to change the plane of the sample and which is helping us to focus the sample properly. So in all the cases you remember that eyepiece, that lens is fixed, only we can change the objective lenses. Okay, what is the key point differences between the electron microscope and light microscope? I know that we didn't discuss about electron microscope too much, but this is a very basic difference between the light microscope and electron microscope. So I am just talking little bit about the electron microscope, so it will help me to discuss about electron microscope in the later stage. So I discuss about this one couple of times, that in light microscope light is using to illuminate the sample, and then in case of electron microscope we are using electron to illuminate the sample. Now what is the key difference here? key difference is the wavelength of the light. What is the wavelength of the light? So, wavelength is playing a very crucial role. I discuss about these things couple of times that okay, we are majorly using glass lenses to focus the light rays. So, glass is a very good, glass lenses are very good to converge or diverge the light, and that is good enough to focus any object properly and get that image. But case of electron, we cannot use glass lens to converge or diverge the electron beam. So, we need a different types of lens system, that is called the magnetic lens. Converging means the beams are coming closer and diverging means they are going far away from each other. So, any magnetic lens we can use it to focus the electron beam. Light microscope, as I mentioned previously, the light wavelength is very large. It is almost 450 nanometer to 750 nanometer. Eye is an wonderful detector for the visible light and it is not damaging. That is the reason for basic biology lab we use light microscope to observe the cell and the large architecture of the cell, viruses, bacteria, like that. So, for the electron microscope we need a specialized sample preparation technique and vacuum environment to operate it. Lower magnification compared to electron microscope. So, for glass lenses we cannot go beyond certain magnification, that we will discuss more when we will discuss about the wavelength of the photon and electron. Can achieve higher magnification, resolution, allowing for the detailed observation of the smaller structure like organelles and the molecules. Now, it will be better to just go through little bit about the glass lenses, how the glass lenses is working, because in electron microscope we are not using the glass lenses, but we are going to use similar kind of ray diagram to explain the image formation. So, that is the reason it is better to go through these simple formulas which is help us to later to explain how image formation is happening in electron microscope. So, this is a lens system and how we can get a focus of any particular lens. So, if we just consider sun is there, when we are at school probably we all played with this lens, right. So, if there is a dry leaf or paper, we are just using a lens system and using the sunlight to burn that paper or dry leaf. So, here we have a lens and we are considering sun is at infinite distance. So, sun rays are coming and all the rays are parallel, and then using this lens we are trying to merge or converge all the lights in one particular point. And if we keep it for long time, it will generate lots of heat and paper or the dry leaf will start burning. So, here when it is forming a small spot, that is basically the focal point of that particular lens, and this is the focal length of that lens. So, here there is a very simple image where we have an object, light rays are coming. So, one straight light, this is the optical axis, this is the optical axis where one straight light is going and forming an image, the image plane. There are three other rays which is deviated its path and then converged in one particular point and giving us an image. So, one of the rays is going through the focus. So, if we have a convex lens, and if we have an object, it will always form a real image, inverted image. Is this statement true? That if there is a convex lens and if we have an object in this particular position and it will form an image, and my statement is that it will always form a real image and image will be bigger than object and image will be inverted. So, this is my question. In next class I will discuss about that part also, but in the meantime you can try to answer this question without checking my next lecture. That will help us in future when we will do our assignment or ray diagram. It will really help us. So, thank you all for your attention, and rest of the part we will discuss in the next class.